In this video, we will be presenting the Ergonome 500, a light optical microscope that is unique and unsurpassed in its high magnification, resolution and depth of field. This short film concentrates on showing specimens taken from the medical world, where direct comparisons have been made between conventional optical microscope methods and the new methods as seen through the new universal microscope Ergonome 500. This advanced microscope allows you to work with both the conventional methods like bright field, dark field, etc., and then simply change over to any of the new methods quickly and easily. This new technology makes it possible for you to view living biological structures at a greater magnification, resolution and depth of field than was previously thought possible. This picture demonstrates the variable depth of field on a measured grating seen at a 30-degree angle. This 3.5mm thick bone slide also demonstrates the tremendous depth of field of the Ergonome 500, which allows you to see all of the solitary trabecular structures throughout the entire depth of this sample. The excellent, well-focused contours, including those in the lower region lying fasciculated bone, is made visible by the high-definition optical construction of this microscope. This species, that belongs to the Thrips family and is mainly a gum tree parasite, is seen here as a young white imagio and also as an adult dark brown winged insect with a skilled chitin skeleton. The capabilities of the Ergonome 500 allow you to see the entire insect sharply, even including the structure of the lower part of the leaf. The finest details of this long-legged fly can now be clearly seen due to the incredible depth of field available through this microscope. The legs and the leg joint fracture section, the haired tentacles, the big compound eyes, the mouth and anal regions, the lateral segmental breathing holes, the vibrating bodies used for flight stabilization, and the wing structures in the finest detail. The following is a comparison of various specimens as seen through conventional microscopes and the Ergonome 500, where special attention should be paid to the improved contour focusing, greater depth of field, the variety of the finest color nuances, and enhanced picture information at both a higher resolution and magnification. This slide shows muscular fibers using a conventional microscope, and now once again as seen through the Ergonome 500. Here you can see a rat's testes under a normal microscope, and again as seen under the Ergonome 500. This is an oesophagus mucus with ciliated epithelium, as usually seen. And now, once again, with the Ergonome 500. With this comparison, you should note that the slide specimen had to be stained in order to be seen under a normal microscope. Using the Ergonome 500, however, such staining is unnecessary and actually disturbs the image. Without staining, you'll be able to see much more. This living heart muscle cell culture shows, at first, the normal rhythmic synchronous contraction and dilation. After provoked irritation, the formation changes into an asynchronous tachycardia and then into a state of fibrillation. Eventually, the cell on the top left dies, followed by the cell on the bottom left. And finally, the cell on the right of the picture ceases its activity. All the cells detach themselves from the cover glass into the nutrient fluid during the course of their deterioration. The whole process is equivalent to an infarction or an in vitro heart attack, and was filmed with help of the ergonome microscope and the Aldrich chamber. This living ciliated epithelium was also taken with the same technique, partly in the complete tissue formation, and partly as a single cell that aimed to bind to the whole formation according to the natural physiological integration, not only in slow motion, but also at normal speed. Ciliated epithelium appears, for example, in the respiratory tract and has the task of transporting foreign particles out of the pharynx. Here you can see into the protoplasm of nerve cells with its different cell organelles. The moving, dark, small balls are small neurotransmitter-filled vesicles. The chemical transmitter substances carried in these vesicles are transported to the nerve contact points called synapses and are emptied there. The vesicles and synapses are jointly responsible for the transfer of nerve impulses. With the ergonome microscope, you can precisely follow the transport of these vesicles into the neurons. In the same way, you can also notice an extracellular transport along the neurons at different points, which up until now has not been described in scientific literature. Here on a monopolar cell, and here on a bipolar cell. In the living culture, you can even observe the contractile ability of nerve cells. These contractile features of the nervous system are mentioned only rarely. Here is a brain section depicted in different planes. Due to the variable regulation of both depth of field and high resolution, the ergonome 500 microscope permits the application of the so-called confocal technique. This procedure, which can be compared to sectional X-rays, allows you to see high-definition images of any layer of a fixed slide specimen, without impairing the sharp definition of either the higher or deeper layers of tissue. The confocal technique has been used to film this mouse, where the fur has been previously removed from the thorax area. As you can see, the heartbeats can be clearly observed through the thorax wall. This technique also allows you to observe the waste elimination movement in this mite. This is the tail of a tadpole of 2 mm in length. The arterial and venous bloodstream can be clearly recognized, as well as the capillary connections. On close observation, you can see that only one single erythrocyte can pass the capillary lumen. White blood cells have the ability of amoeboid locomotion using pseudofeet, the so-called pseudopodium. This allows them to engulf foreign bodies and bacteria into themselves and neutralize them. Here, you can see exactly how this phagocyte moves. This active living white blood cell uses endocytosis to engulf the endogliotic particles located in the serum and transports them from the marginal region into the center. With increasing migration towards the center, the engulfed particles lose their activity. In this stained slide specimen,